Okay, welcome, my biochemistry friends. Um, we're gonna talk about glycolysis, but we have to put in a little bit of work before we get to that point. So I'm gonna do some kind of setup for the rest of the semester here. And we have to talk a little bit about how metabolism works and what the delta G dynamics are. We're going to do just a brief thing about what sugars, the structure of sugar, and then we'll start going into glycolysis. And I'll remind you that we are now kind of talking about catabolic processes. And in catabolic processes, we are taking something that's complex and making it more simple, and in the process, we're pulling energy out of that, and we capture that energy in the form of ATP or reduced electron carriers like NADH, NADPH, or FADH2. On the flip side, we then use that energy to take things that are simple and make them more complex. Uh, so catabolic processes tend to be exergonic. They have negative delta Gs, and they are used to drive anabolic processes that have these positive delta Gs. And so the energy of ATP, NADH, etc., are used to drive anabolic processes. You form the low energy counterparts to each of those that get recharged by catabolic processes. This is why we eat, right? Uh, so that we can constantly have that source of energy going forward. And as I've said several times this semester, the catabolic processes are convergent, and they converge on these acetyl-CoA units. Um, Whereas anabolic projects are divergent, and they diverge from these acetyl-CoA units. So from the acetyl-CoA units and really from the citric acid cycle, you can get all of these biological macromolecules. Um, and we break down all these different biological macromolecules molecules to form these acetyl-CoAs. And so this is kind of a topic of an advanced biochemistry class, but I will say a couple of things. We can define flux of these metabolic processes, and that's simply the flow, the input and output of the atoms that are going through here. So how quickly do we go through these processes? And that's a function of the regulation and cellular level, the protein level, and uh, things of that nature. Now we exist in a state of dynamic steady state, right? And so that should be kind of contradictory, right? Steady state means being consistent, but dynamic means change. And so we are in this state in which we're actually throwing atoms into the system constantly, but the steady state concentrations of the intermediates don't change. Even though we're constantly adding stuff, we're also pulling it away at the same time. So that's the influx and efflux that you see here. And again, here is a small metabolic pathway, right? And then this is what we're kind of discussing now in the citric acid cycle and glycolysis here. And you see that that fits into all sorts of things. I think, for instance, I believe that this is beta oxidation of fatty acids here. This is the urea cycle, so this deals with amino acid catabolism. And you can see each of these little dots are are enzymes and steps in metabolic pathways. And so you can see how interconnected these things are. Um, and again, they are related. So what we're going to do is talk about glyco glucose metabolism, but that's just kind of one way to get here. There are all sorts of entrees into this pattern process. So if you take lipids, lipids are triglycerides, right? So they have a glycerol head and then they have these fatty acid tails. You actually separate those two. Um, in beta oxidation fatty acids. The glycerol looks like a three carbon carbohydrate, and so that feeds into the glycolysis pathway. And then the fatty acids are broken down to form acetyl CoA units directly that go right into the Krebs cycle. Um, Similar mechanisms occur for amino acids where you can chop off part of it. The amine group is excreted as a waste, right? But you can chop off parts of it and they feed into various places within this pathway. And so from this you can get into you can form the acetyl CoA units that are used to make this energy. And so here, this is the pathway we're going to spend time on: glycolysis, citric acid cycle, and oxidative phosphorylation. Obviously, there are others. Plant biology, you'd go deep in carbon fixation and photosynthesis. In an advanced biochemistry course, we talk a little bit about amino acid biosynthesis and catabolism. There is, I don't go into detail about this, but the biosynthetic pathways, gluconeogenesis, pentose phosphate pathway, are all kind of related as well. And then we might talk about the beta oxidation fatty acid, just depending on time. And all of this is kind of centered on our old friend, the delta G. Um, And so, if we go back to our first principles, we know we can define delta G prime that is related to the KEQ, and that the delta G is negative, it's an exergonic reaction. And the way that we drive endergonic reactions is by coupling it to an exergonic reaction. And as long as there is a net negative delta G in coupled reactions, then you're good to go. And so the way we do biosynthetic reactions is to couple it with the exergonic reaction. Now, we get to do a little bit of quantitation here because there's something that I have to spell out here, and that deals with the actual delta G. All right, we're going to see this is going to be a trick that the cell exploits quite a bit. Now we define a delta G prime, right? And this is standard conditions. And remember, with standard conditions for this delta G prime, it's what? It's something like 25 degrees centigrade, 1 atm, pressure, um, pH in biochemistry terms is 7.0. But more importantly, the standard delta G says that the concentration of the reactants is equal to the concentration of the products, right? And so remember, the delta G in standard conditions say that the reactants and the products must be equivalent to each other, and then it analyzes the energy that drives the reaction in one way or the other. The KEQ says what? That the delta G is zero, how much products or substrate are there at that delta G, right? And so they're related in that way. Now, in practice, in the cell, it is rare that the concentrations of substrate or products are equal to each other, right? And that change in the concentration has an energetic effect. And so we can define an actual delta G as the standard delta G plus an expression that relates to the actual concentration of products um, and reactants, right? And so we call that the mass action or Q. And so here the actual delta G is equal to delta G prime plus RT times the natural log of the actual concentration of these things. And so the cell manipulates this quite a bit. They can take delta Gs that are typically endergonic, and then by manipulating the concentrations of the products and um, reactant can drive it to become exergonic. And so here's an example. This is glucose 6 phosphate to fructose 6 phosphate, so an isomerization reaction. The standard delta G is positive 1.7 kilojoules per mole. But when you factor in the concentrations of fructose and glucose in the cell, then you can see using this expression that the new actual delta G is um, negative 2.9 kilojoules per mole. And so you took a reaction that is endergonic, and in the actual conditions in the cell made it exergonic. Uh, and so this is a way in which you can get to a reaction without having to invest energy in the form of coupling that reaction to something else. So we will see, and I'll say this is one of my favorite questions in biochemistry. Um, After the third step of glycolysis, in which we invest in ATP to form 1,6-fructose bisphosphate, uh, that is the last time we put energy into the system, right? Every other step after that is one in which you extract energy from those molecules. Now, there are endergonic steps, and somehow the cell has to get through those processes. And oftentimes they do that by manipulating the concentration products and reactants. And so if you can see in this, the way to do that is to keep the concentration of products artificially low somehow. Very important concept. I would kind of go back and repeat those last 2 minutes if you need to. All right. Another way to get energy out is from ATP, and we'll look at kind of how we get that energy from ATP. So this is a nice little phosphate group here, pretty strong negative charge. Um, in GBio, we say that the energy from ATP hydrolysis comes from the release of these kind of packed together negative charges. So here you have these negative charges that are all shoved together. They don't want to be there, and so by chopping them, you push off the ATP, and that is true. Uh, Certainly, the bond lengths of these phosphates in ADP are longer than that of ATP, and that is where you get most of the energy. But it's slightly more complicated than that. And I like this kind of concept a lot. And so you do get the charge separation in the products, but you also get some additional kind of bang for your buck in the hydrolysis of ATP. And all this contributes to the amount of energy that is released from that hydrolysis. So here you have the hydrolysis of this phosphate bond over here. And so you release energy from the hydrolysis, and you get a relief of the charge repulsion. So instead of having three negative charges very close together, you have two. And um, they they aren't quite as sick of each other, quarantined together in a small space or whatever. So that is one way to get energy out. But you also get another thing. The phosphate that's released, whoops, gets resonance stabilized, right? And so you have resonance stabilization of this phosphate. That's going to release a little bit of energy. And then the ADP that's released gets ionized, and that's going to have a stability effect as well. And so you have energy that comes out not only from the hydrolysis, but also from the resonance stabilization and ionization of the resultant products that are going to add a little more bang for the buck. So it isn't just that you have that those three charges close together, but also these other things that all contribute to the amount of energy that you get from the hydrolysis of ATP. This is one of the reasons why ATP hydrolysis is such a useful tool for the cell to drive these processes. And so this is what we kind of look at here. If we have ATP at the center of this, we either are going to get ATP by substrate level phosphorylation, by taking higher energy compounds. This is a delta G scale, right? So 1,3-bisphosphoglycerate or phosphoenolpyruvate, these are higher energy compounds in ATP, and then using that energy to make ATP from ADP, and then the ATP itself can be used, it is of higher energy than a glucose 6 phosphate to phosphorylate glucose or glucose 6 phosphate in order to go into the glycolysis process. All right. And so this is how we generate that ATP in order to do the work. And I did want to kind of point out here with the glucose, we're looking at a compound that's fairly heavily reduced. Remember, the reduction state kind of corresponds with the energy level because it has high energy electrons associated with it. And so we look, this is just a few compounds that are organized by their oxidation state, right? And here, carbon dioxide is the most oxidized and has the least amount of energy in the form of these high energy electrons. Here, these alkanes are highly reduced, right? And so the energy is represented by these hydrocarbon bonds. And so if I were to ask, for you to organize this according to its oxidation state or its energy levels, uh, really all you need to do is look to see how many oxygens are present and how many hydrocarbon bonds are present, right? So this alkane is going to be the most reduced because it has three hydrocarbon bonds here, right? This alcohol has two hydrocarbon bonds and then another hydrocarbon bond that's connected by this alcohol, right? Whereas this aldehyde only has one, and then this carboxylic acid has one that is going through this ethanol, right? Carbon dioxide is completely oxidized. There's no hydrogens there at all, right? And what do we do with carbon dioxide? How do we use it to extract energy? Well, we don't, right? It is a waste product in cellular respiration. So there's a little energy in there that we excrete it out as as waste. And this is just a look at those sugars. I'm not going to go deep into carbohydrate structure because I do want to get into glycolysis. But we know that carbohydrates have there is a water molecule, H2O, that's associated for every carbon. So they are hydrates of carbon. And so it is CnH2nO in terms of the ratio. So if you have five carbons, it would be 5:10 to 5. If you have six carbons, it's 6:12 to 6. Um, Here for glucose, one, two, three, four, five, six carbons, right? Fructose has one, two, three, four, five, six carbons as well. So those are isomers of each other, right? Glucose has its carbonyl group at the end, so it's an aldehyde. Fructose has it in the embedded within the structure. So it is a ketone. I'm sure you remember that from organic chemistry. And this has an effect on the ring structure. So glucose forms a six-sided ring. It's carbon number six here, but it forms a six-sided ring, whereas fructose forms a five-sided ring because of the nature of that, where the carbonyl is. Now, note they still have six carbons. Fructose is not a five carbon sugar, it still has six carbons. It's just that the ring structure is different in that case. We'll see this has profound implications in glycolysis. Um, um, I'll say this. There are these sugars are all stereoisomers of each other, and they have kind of two forms, the L form and the D form. And the way to look at that is to count the last chiral carbon, um, the furthest one away from the carbonyl group. And then you look at the position of this hydroxyl, and that will determine whether you're in the L form or the T form. So this is D because here is the carbonyl group. Here is the last chiral carbon. Carbon number six isn't chiral, right? And then the hydroxyl is on the right. So it's going to be in the D form. If this was flipped, it would be the L form. And I think in organic, you guys get into RMS structures and stuff like that. We don't do that. And so here, if you have isomers that differ only in the position of one of these hydroxyls, they're called epimers. So glucose and mannose are the same, except one of these is flipped. Um, and so those are epimers of each other. If you have more, then they aren't. Um, and again, I'm not going to get into the too deep into the kind of chemistry of these because I do want to get into glycolysis. But if you look at what happens, so this is the regular form of glucose, but they form these rings. And the way they form these rings is that this hydroxyl will link, don't do that, will link up with this um, carbonyl group. Now, when that happens, you actually form a new chiral carbon and call that the anomeric carbon. And then, depending on the orientation of the hydroxyl that is formed here, if it's in the downward position, it's an alpha. So, this is a D glucose, right? So, it's D. Um, Glucose, it is in that ring structure, so it's pyranose. And then if it is down, if the hydroxyl is in the down position, it is alpha. And if it's in the up position, it is beta. Right? And so you now formed a new carbon. This is the anomeric carbon, and this is actually the place where chemistry can occur on these glucose molecules. And so when you form macromolecule structures, you form a glycosidic bond between this anomeric carbon and some part of another um, uh, carbohydrate. And then depending on the nature of these, they can have profound implications on the nature of a sugar. So whether you're in alpha um, or beta form really determines whether humans can break down those sugars or not. Um, so a lot of good chemistry that's there, but again, I'm going to skip through a good portion of it. Um, and this the breaking of this bond, so lactose is a disaccharide, so it has two of these glucose and galactose. Whether we can break this bond, there's an enzyme called lactase that does that. We all have that enzyme um, as a infant, and then most of us tend to lose it as we go into adulthood because there's no reason to digest milk. Um, there are a couple of populations that spontaneously evolve the persistence of lactase um, in adulthood. I think there's a Maasai population in Kenya and then something from the Baltics or something like that. But for most of us, unless we have that enzyme in adulthood, we lose it and we lose the ability to break the glycosidic bond in lactose. If we don't have the ability to break that bond, that sugar stays in our digestive systems, but bacteria eat it up and love it. And so, you leave all that energy, you aren't extracting it yourself. The bacteria is tearing into it and growing like gangbusters and producing a lot of, you know, gas and things like that. And so, for those of us who are lactose intolerant, it is because we lack this enzyme. And so we can take something like Lactaid, which has pre-broken down the lactose. Uh, so that we can extract that energy out and bacteria doesn't have it, or obviously just avoid dairy, right? Okay, so I think that that is enough to get us to kind of look at breaking down the sugar. This is the general process of glycolysis. Obviously, we're going to go into a ridiculous amount of detail, but we're splitting glycolysis. So we are lysing a glucose molecule, taking one 6 carbon structure and doing it into two 3 carbon pyruvates. 10 steps to do that. Half of it is a preparation or investment step where we are spending cost 2 ATP, and then half of it is a payoff step in which you release 4 ATP. So we're going to get a net of 2 ATP out of this process. No oxygen is needed, and this occurs in the cytoplasm. This pyruvate that's generated, oh, we also get some NADH here. So we reduce the electron carrier NAD plus to form NADH. And the pyruvate can go on to be broken down in the presence of oxygen. This goes to the citric acid cycle. So for glycolysis, you have one molecule of glucose, you have 2 molecules of NAD plus, ADP, inorganic phosphate. You form pyruvate, NADH, ATP, plus a little bit of water and some protons here. Note, if we're counting the carbons, 6 carbons of glucose, 3 carbons of pyruvate. Oops, yeah, 3 carbons in pyruvate. So that's that process. That glycolysis process, then the pyruvate will go in cellular respiration into the mitochondria in the presence of oxygen. It'll be broken down via the citric acid cycle, and then the electrons will go into the electron transport chain to form ATP. Um, I don't know, where's ATP here? To form ATP from ADP and inorganic phosphate via oxidative phosphorylation. So we're talking about the glycolysis process now, looking at the enzymes that are involved in that, but then we'll get into the citric acid cycle and oxidative phosphorylation. Fun stuff. And this is the chemistry of what's going on. We have this glucose, the presence of oxygen, gets completely oxidized to carbon dioxide. So you're taking something that's reduced and oxidizing it. oxidizing. Oops, there's no T. D. And then here you're taking something that's oxidizing and reducing it. In the process, you're forming this energy. Uh, if you were to completely oxidize it, like just burn it up, you get 686 kilocalories per mole. We aren't going to capture all of that. We'll only capture about 25% of it by the time all is said and done. And the way we capture it is in the form of ATP. And again, if we put this in context of kind of total energy dynamics, the source of all of our energy is the sun. Photosynthetic organisms translate that energy of the sun into chemical energy in the form of sugars, glucose, and then we undergo glycolysis. This is something that every organism does to form this pyruvate. In the absence of oxygen, you have an anaerobic process, fermentation, and you get the two molecules of ATP out of it, and that's it. In the presence of oxygen, much more efficient, you can get up to 32 molecules of ATP simply by having that oxygen available and going through the complete cellular respiration process. And so that's shown here. Cellular respiration, when oxygen is present in animals, the final electron acceptor is oxygen, and then the electron ends up in water. Without oxygen in animals, the final electron acceptor is pyruvate, and then that gets converted into lactic acid, which can ultimately be regenerated later on. If we have an organism like yeast, the final electron acceptor is acetaldehyde, and then that gets converted into ethanol and is excreted from the system. This is a process called fermentation, which gives many of the compounds that we know and love. Um, yeah. And so this is the total process. We have, blow this up. Yeah. The glycolysis that we're going to talk about in this chapter, you get 2 ATP out of that. You go through the production of acetyl-CoA, so you take this 3-carbon pyruvate and form a 2-carbon acetyl group. You generate a little carbon dioxide in that process, and then this acetyl group feeds into the Krebs cycle. Here, you generate all of the remaining carbon dioxide, so you've broken down the sugar by this point. You generate 2 molecules of ATP, so just a little bit of ATP, but you have lots of these reduced electron carriers, and then we're going to use the electrons to drive a proton gradient across the inner membrane of the mitochondria that forms lots of ATP. And in the process, this electron gets taken up by the oxygen that we breathe in to form water. And so that's what we're going to spend the next few weeks going on. I think last piece of bean counting, and then I think we're ready to get into it. And just talking about oxidation reduction reactions, I think we've already done this, but remember, we're looking at the flow of these electrons. And so something's reduced if it has the electrons, it passes those electrons to something that doesn't have them. That's the oxidized compound, right? And so here, these electrons are passed from A to B. A has become oxidized, B has become reduced, and this guy is where the energy is. And so you can pass these electrons on directly, or you can pass them on in the form of hydrogen atoms via electron carriers. So here you have this hydrogen atom. It gets passed to NAD plus to form NADH. This is where the energy is. So now it is oxidized here, NADH is reduced, and NADH will pass it on to compound B. So now this is the energy here. So these NADH is an electron carrier because this job is simply to pick up electrons and then shove them off someplace else. And it just cycles between this oxidized form and the reduced form. Why am I keep on doing the oxidized? What am I doing? And reduced. And then this is a look at this. Here is the group, the nicotinamide group that actually gets oxidized, and you can kind of see in the oxidized form you have the N plus here. In the reduced form, you have a hydrogen atom that's over there. The electron is in this nitrogen, and then you actually have a proton that's kind of floating around there associated with it. So it's picked up a pair of electrons in this process. And this NAD, as you can see, has a nitrogen containing base, a ribose sugar, and phosphate group. So this is a nucleic acid. Nucleic acid. All right, so this is just a modified nucleic acid. Instead of that third phosphate group, we have this nicotinamide group. And there's a lot of energy here. The oxidation of NADH in the presence of oxygen yields 52 kilocalories per mole. Remember, ATP hydrolysis yields about like 7 or something like that. It's a lot of energy in these NADH molecules. All right, so now we've kind of done the setup so that we can do glycolysis, and I'll do that in the next lecture.